Yeah, hello and welcome to today's Mycotalks. It's a pleasure for me to welcome you here together with my co-host Gordon Brown. I'm sure that all of you by now know Mycotalks, but despite this, I'm going to say a few introductory words before we hand over to our speakers. Now, today we have a program with two talks. One is about carnivorous fungi and their interaction with C. elegans, given by Ping from Taiwan. And the second talk will be by Kwon-Chung from the NIH, and she's going to talk about differences between Cryptococcus neoformans and Cryptococcus gattii. So as you know, Mycotalks is a seminar series that is organized by the MRC Center for Medical Mycology at the University of Exeter. And you can also, and I encourage you to do so, follow them on Twitter. And there is an organizing committee that tries to select cutting-edge talks on topics around medical mycology. Of course, the topics and the quality of the science is in the center of being selected as a speaker, but we also focus on career stage, on gender and diversity issues, and try to be representative for the whole fields. We will have two talks today, as in all sessions, and then an in-depth discussion. And already now, I would like to encourage you to submit your questions using the question and answer function. And please do so already during the talks. Gordon and myself will then collect all the questions, and in the end, we have a discussion. For those of you who are not here, you can watch all the talks in the seminar series on YouTube, and all the past talks are also there. We will have more talks this year to come, of course. On the 25th of May, the next edition will take place, and the speakers are George Kamilos from Crete and Anuradha Chowdhury from Delhi in India. And then in June, we will have a scheduled meeting on the 29th of June with Agostinho Carvalho from Portugal and Xiao Lin from Georgia. Now, due to the success, I guess, of Mycotalks, there are also now sister series of different talks, and we have a seminar series focusing on clinical talks, the Mycoclinics, and the next one will take place on the 8th of June with Paul Favey and Neil Clancy about challenges in antifungal drug resistance. And there is also a seminar series for trainees, the Medical Mycology Trainee Seminar Series. And we encourage you to get informed about this as well, and also suggest speakers for this seminar series, or suggest yourself as a speaker for the trainee seminar. So with this, I'll end my introduction and hand over to Gordon to introduce our first speaker.
Thank you very much, Oliver. And just to re-emphasize what Oliver said about please putting your questions into the Q&A box at the bottom of your Zoom screen. And then just one addition, just to add the name of the speaker whom that question is addressed to. So it's my great pleasure to introduce Professor Yingping Shi, who's based at the Institute of Molecular Biology at Academia Sinica in Taiwan. It's very nice that Ping has agreed to give a talk here. It's 11 o'clock in the evening for her, so it's almost bedtime. So she's agreed to stay awake and give us a talk, which is very nice, as I said. So Ping did her PhD and first postdoc at Duke University. Then she did a postdoc at Caltech before moving to Taiwan in 2015, where she's been based ever since. Since establishing her own laboratory, Ping has been highly prolific, and the excellence of her research has led to numerous awards and honors, including the Taiwan Outstanding Woman in Science Award, which she was awarded last year. So Ping's research interest focuses on the interactions and co-evolution between the nematode, C. elegans, and nematophagous fungi as a model to study interspecies communication and predator-prey co-evolution. And as you heard from Oliver's introduction of her title, she describes predatorial fungi as carnivorous, which has sparked the interest of the media. And her work's recently been highlighted in the New York Times and in Scientific American. And carnivorous fungi is what we're going to be hearing about today. So thank you again, Ping, and over to you.
Okay. Hi, everyone, and thank Gordon for the kind introduction. Good morning or good afternoon, And okay, let me see if I can share my slides.
You're good, Ping. That's all good.
Okay. All right. Yeah. So, okay.
Ping, you've gone back to presenter mode. You want to? Yeah, there you go. You're good.
No, okay. All right. All right. Thank you, everyone. And today I'm very excited to be here and to tell you about our recent work. And due to time limit, I will be mostly focusing on the oyster mushroom for today's talk. So we know that the model organisms have contributed enormously to all domains of biology. So these organisms are really arguably probably the best understood organisms on this planet. However, it's kind of surprising that till today, if we look into the genome database for the budding yeast, we still only know the function of the genes in the genome, about like 2/3 of the genes. But and in the nematode C. elegans, this number dropped to about like 50%. So because I think these model organisms are largely studied under a very well-controlled laboratory settings, and which is very different from their natural environment. So I thought that if we can try to understand the natural history of these model organisms, and we might be able to learn more about them. And I think C. elegans is a great model to ask these questions. And among the various biological interaction that the C. elegans encountered in the natural environment, I'm mostly interested in their interaction with their natural predators, which includes many group of fungi. So why am I interested in predator-prey interaction? Because I think this interaction is really universal. It occurs across the tree of life, and not only in the kingdom of animal, but in other kingdoms, we also find many examples of predator-prey interactions. And these type of interaction really often resulted in the coevolution between the species and contribute to many of the amazing adaptation that we observe in nature. However, these interactions is difficult to study. So I thought that C. elegans will provide a unique chance for us to address this question at the molecular level. And then we turn to the predator, which are the carnivorous fungi, okay? And these carnivorous fungi have independently evolved across all major phylum. So it's been known for a very long time that some fungi have evolved the ability to prey on nematodes. And one of the reasons is that these nematodes are the most abundant animals by number on this planet. So many of the fungal species, they evolve the ability to prey on them when they are under starvation. In my laboratory study, like two groups of carnivorous fungi, and one is better studied nematode trapping fungi, and the other is the delicious oyster mushroom, Pleurotus. And they evolve very different kind of mechanism. So today I will be talking about mostly how Pleurotus oyster mushroom paralyze and kill the nematodes. So let's watch like a short video about how these fungi prey on C. elegans. Okay, so apart from the amazing biology for this carnivorous fungi that intrigued my curiosity to study them, one other reason for practical application is that there are many nematodes, they are parasites, and we currently have very limited ways to control this parasitic nematode. So I thought that by better understanding the biology of this carnivorous fungi, maybe one day we can harness the nematocytal potential for these fungi and try to find the ways to control these parasitic nematodes. Okay, so the story really begins when I came across this paper published by Greg Thorne and George Barron back in 1984, where they reported that pleurotus are carnivorous. Okay, so they can paralyze nematode and they kill the nematodes. But then there's still, like, after the report, very little is known about how this mushroom exactly kill the nematode at the molecular level. So I thought that this is an interesting problem, and we decided to try to understand this question in further detail. So the first experiment was really like we went to the supermarket, get the oyster mushroom, and then grew in the lab. And then we put C. elegans on this mushroom culture and see what does C. elegans behave. And as you can see here, like these are the fungal hyphae of the pleurotus ostreatus. And once the C. elegans touch the fungal hyphae, within like a minute, the nematode is quickly paralyzed. Okay, so this response is very dramatic and very fast, comparing to its response to nematode trapping fungi. So we thought that, yeah, it's really interesting, and we really want to understand how this nematode get paralyzed so quickly in more detail. So we look into like a different genus of pleurotus and to see different species, like of the pleurotus, and see whether they can paralyze C. elegans. And what we observed is that indeed all the pleurotus species paralyze C. elegans. And then we also look into whether the oyster mushroom can paralyze a wide diversity of nematode. And indeed, like all the nematodes we could obtain and grew in the lab, and we test them, and they are all paralyzed by the oyster mushroom. So this result tells us this predator-prey interaction is highly conserved. So my first PhD student, Jing Han, who is currently a postdoc at the NYU now, decided to carry out the forward genetic screen. We try to isolate the C. elegans mutant that can become resistant to pleurotus. So we did a very thorough screening, and then we did isolate many C. elegans mutant. As you can see here in this video, it became resistant to pleurotus, and it can move freely on the fungal hyphae. So then we use whole genome sequencing and also genetic mapping to narrow down the mutation in each of the mutant. And we found that basically all the mutant, they have a defect in genes involved in ciliogenesis. So cilia are the nerve ending of the sensory neuron in C. elegans. As you can see here, it's the tip of the neuron. And for this structure, it's very important for the neuron to sense the environment. So this result suggests that somehow the pleurotus is attacking C. elegans from the sensory cilia. So if the C. elegans lack this structure, it cannot develop cilia, then it can become resistant to pleurotus. So we did some more detailed analysis. For example, we put these gene back in the mutant, and it can rescue the resistant phenotype, and the worm would become paralyzed again. And also to determine whether it's the cilia structure itself is important for this resistant phenotype, or it's the signaling function. We tested many C. elegans mutant, And from here, we conclude that it's only when the cilia structure fail to develop, then the C. elegans mutant can become resistant, suggesting that the cilia structure is important for the fungal toxin to exert its activity into the nematodes. So exactly what happened to the neurons once the C. elegans touch the fungal hyphae? We use GFP labeled neurons strain and then test its morphology. And as you can see now in this figure, when C. elegans touch Pleurotus, very quickly, this image was taken like 5 to 10 minutes after the worm exposed to the Pleurotus hyphae. And we observed that all the neurons are undergoing like a very obvious necrosis. And this phenotype is not just restricted in the sensory cilia and the sensory neurons. So gradually we observe that basically all the different tissues in nematodes start dying, okay? So this includes like a different kind of neurons and in the muscles and basically all tissues we have tested. We observe very rapid cell death in the entire nematode. And this mechanism is distinct from all the nematicides that currently are used. So aldicarb, levamisole, and ivermectin, they function at the silencing the neuromuscular junction, but they do not directly cause cell death. So we thought that while like Pleurotus is killing the nematode from a very different mechanism. So, but to really understand exactly how Pleurotus kill nematode, we really need to know the identity of the chemical. Okay, when we saw the identity of the chemical of the toxin, we cannot really pin down the molecular mechanism. So exactly what is the toxin? Okay, so there is a report back in 1992 published in Journal of Chemical Ecology reporting that this trans-2-decyanoic acid is the toxin that produced by Pleurotus ostreatus. But when I read this paper, I thought that maybe there should be other compound involved because we know that the Pleurotus only produce this potent toxin under starvation condition, okay? But then in this paper, the Pleurotus, the toxin was isolated from a culture that's very rich in the nutrient condition. So we have our chemist collaborator, Dr. Junjie Hsieh from Seneca in the Institute of Chemistry, synthesized this compound for us. And then we treat C. elegans with this compound and found that it doesn't trigger the typical cell necrosis phenotype. So this results indeed suggest that there are maybe additional, the major toxin remain to be discovered. So we try initially with a chemical approach and try to extract the hyphae and want to purify whether there's from the crude extract, can we try to identify the toxin? But it was never successful. Like all the crude extract we obtained just do not have any biological activity. So I felt that while trained as a geneticist, we would like to use a genetic approach in Pleurotus again. So very lucky, I met Dr. Yoichi Handa at the Kyoto University in a European fungal genetics meeting in March 2018. And he talked about his work in Pleurotus and mentioned that his lab can do a forward genetic screen. So he very kindly hosted me and two of my students to visit his lab, where we learned how to carry out mutagenesis. And then that summer, we came back to the lab, recruited many undergraduate students, including Yiyun, who was an undergraduate student back then, and we started doing a genetic screen to try to isolate the Pleurotus mutants. So we screen many Pleurotus mutagenize the clone. And we screen like basically 2 summers. And eventually we screen like 12,000 clones and identify about like 20 mutant. We call them lot mutants, indicating a loss of toxicity for nematodes. And this is one of the mutant of Pleurotus that we obtained from the screen. And as you can see now, like now these mutants, they failed to paralyze C. elegans. So worm can move happily on this mutant Pleurotus. So we have all this about like 13 mutants that exhibit very strong phenotype. Basically, they completely lost any toxicity to C. elegans. Okay. And so we decided to look in more detail about this mutant. And we found that actually the wild type Pleurotus hyphae, there are lots of lollipop-like structure. And previous report nematode on these Pleurotus hyphae. And interestingly, all the lot mutant we identified from the genetic screen completely failed to develop this structure. All of these mutants do not have this structure. Okay, so this result suggests that maybe the toxin that we are looking for is specifically like inside this lollipop-like structure. And it's interesting because these structures are very fragile. So if we touch it, it just breaks right away. And when nematode touch the hyphae, okay, it also just disappeared. So we did a very simple experiment. So the wild type hyphae contains lots of this lollipop, and we just basically add some glass bead on the plate, and we just shake briefly for a few seconds, and these lollipops were gone. Then immediately we put C. elegans back on this fungal culture. And this is what we observe, okay? So basically a wild type hyphae, if we shake with glass bead, okay, the toxicity is immediately lost. And this result gave us a hint that maybe the toxin might be volatile in nature, so that it's instantaneously became non-toxic to C. elegans. So having this hypothesis, we decided to carry out the GCMS analysis on Pleurotus, and we designed like 3 conditions to try to identify the volatile from this lollipop. So in the first condition, it will be just the media and the glass bead. And in the second condition is media, and we inoculated the Pleurotus ostreatus. And the third condition is the media, we inoculate the Pleurotus ostreatus, but we also added some glass bead inside the bottle. So we let the fungus grow for a few weeks, and so that it covered the entire bottle. And then when we're ready to sample the headspace, under this condition, we shake the bottle so that all the toxocysts are broken. And we immediately sample the headspace with spinning fiber and send for GC analysis. And specifically, we are looking for if there's any compound that can be detected in the shaken condition. And sure enough, we very repetitively identify one compound in this condition, which is 3-octanol. And we decided to just get this compound. It's available in sigma and tested whether it has any activity to C. elegans. And we found that it basically recapitulates all the phenotype we observed, like when we put C. elegans on the fungal hyphae, it paralyzed the nematode above certain threshold completely 100%. It causes rapid neuronal cell death. It triggers massive calcium influx in the muscles. Okay, so these are the 3 major phenotypes we observe when C. elegans encountered Pleurotus hyphae. So these results suggest that the 3-octanol is likely the major, one of the major compounds that causes the paralysis and death phenotype in C. elegans. So, but exactly how does 3-octanol, such a simple compound, how can this compound do it? So we decided to further study the molecular mechanism. So we first did some TM to look into the C. elegans tissue more carefully. And we found that basically once C. elegans touch pleurotus or encounter 3-octanol, very fast, just a few minutes, immediately the mitochondria in the muscle cells was extremely swollen. Okay, so you can see this is the normal mitochondria, and this is mitochondria after C. elegans touch pleurotus hyphae or 3-octanol. And if we use a calcium indicator expressed in the mitochondria, we also observe a massive calcium influx into the mitochondria. And this suggests that the calcium influx causes the mitochondria swollen. So we use reporter line and just check mitochondria morphology in different tissues in C. elegans. And as you can see, this is the mitochondria in neuron or in the hypodermis cells. And you can see that when the nematodes touch pleurotus or 3-octanol, like all the mitochondria just swollen, okay, and many of them are disrupted. And how exactly these 3-octanol can trigger such a rapid, massive calcium influx? We hypothesize that maybe it caused some membrane damage, because the extracellular calcium level is really much higher than intracellular. So if the membrane is damaged, then immediately it will cause lots of calcium influx. So we use some reporter line, which labels the plasma membrane in C. elegans tissues, okay? And this is the hypodermis tissue. And as you can see, this is the wild type C. elegans without touching the pleurotus hyphae. Once the nematode is touched the pleurotus hyphae or encounter 3-octanol, you can see that the integrity of the plasma membrane was disrupted. Okay, so this was like 5 minutes post exposure to the fungal hyphae of the compound. So, and if we use in vitro liposome, then we also observe that the liposome morphology get altered very dramatically. And furthermore, we observe an interesting calcium wave propagation in the nematode once C. elegans touch the fungal hyphae. So the toxin will get into the nematodes through its nose from the sensory cilia. But gradually we can see this wave of calcium propagating from the head to the tail in the hypodermis tissue in C. elegans. And this wave of the calcium propagation will lead to the propagation of the cell death. So if we scan the dead cell with perpethium iodine, and we can see that after touching pleurotus hyphae, we can gradually see more cells start dying, okay, from the head to the body. And gradually, like an entire nematode, there are many dying cells. And to further understand, like how is this wave of calcium can propagate in C. elegans, and what could be the mechanism that control this propagation? We tested a few mutants, and we had a few different hypotheses. And basically what we found is that the membrane fluidity is quite important. So if we use C. elegans mutant that has more saturated fatty acid on their plasma membrane, then we can see that the propagation of the calcium wave get slowed down. And you can see that this calcium will be more restricted in the head region. And in this mutant, we also observe that the dying cell, okay, will also be more restricted in the head region. Then lastly, we are curious about whether other structurally related compound to 3-octanol can also kill nematodes, okay? And so we tested other ketones that different ketone with different lengths of the carbon chain. And what we found is that the position of the ketone doesn't really play a major role in terms of their toxicity to nematode, However, the carbon chain, okay, the length of the carbon chain is very important. So 3-octanol is like sufficient to trigger cell death. But if we use 6-carbon ketone like hexanol, it doesn't really have much toxicity to nematode. But if we use like a decanol, okay, so the carbon chain is longer, then this compound is actually more toxic to nematodes. So suggesting that the exactly like how the more hydrophobic the compound is, maybe it will like disrupt the membrane integrity of the C. elegans more efficiently. Okay, so basically, yeah, our work suggests that the pleurotus have evolved a strategy that we call it's like nerve gas on a lollipop to kill the nematode. So they develop this structure, toxocyst, that allow them to concentrate the 3-octanol to a very high local concentration. And when nematode touch this 3-octanol, this 3-octanol get released and get into the nematode cell from the sensory cilia and trigger the cell death and a wave of calcium, and it propagated inside the nematodes. Okay, so now we are really curious about what's the mechanism of the development of toxocyst. How does pleurotus generate this interesting structure? So as you know, like we have did a genetic screen, so now we have lots of these LA mutants. And so we want to like narrow down like the mutation in this LA mutant. So the mutagenesis was carried out by UV, so it has lots of random mutations. So first we assemble a high quality genome of the PC9, which is the wild type. And then we conduct whole genome sequencing of this LA mutant and identify all the potential mutations. Then we also try to use genetic mapping to narrow down the candidate mutation. For example, we cross it to a different strain background, okay? And then we dissect the progeny and use SNP mapping to narrow down the candidate mutation. And from this strategy, we can identify a couple like candidate genes. And now we are in the process of trying to pinpoint the exact like a mutation in each of the mutant. And on the other hand, we also want to like get the genetic system more established in pleurotus. And so we think that it's important to have a congenic strain pair so that we can really do a lot of genetic analysis. So PC15 is a genetic mating type compatible strain, but it has very different genetic background. So we have conducted a series of 10 back crosses and then generate a congenic strain for PC9. Okay, as you can see now, this congenic strain, it has a good growth, like it grow well. And then it's also mating type compatible with the PC9, okay. So we use nanopore to assemble a high quality genome of PC9.15, which is the congenic strain. And then now we plan to use this congenic strain to set up a lot of bulk segregant analysis and using pool sequencing to identify all the like candidate mutations in these lot mutants. And the goal is, to try to figure out exactly how toxocysts develop in pleurotus, because we think the evolution of this structure is really contributing to the evolution of carnivorism in basidiomyces. Because all the species that we observe that can paralyze nematode, okay, they can hunt nematode, they basically all develop this structure. And if we go to species that are further away, okay, that do not paralyze nematode, they are not carnivorous and they do not develop this structure, okay. So so now we're trying to understand how this structure is developed in pleurotus. And lastly, I want to thank the people like in my lab who did the work. So this work is mainly like carried out by two of the graduate students, Jinghan and Yun, okay. And then I also want to thank many of my collaborators. And for the project I talk about today specifically, like we thank Dr. Honda for teaching us how to carry out the mutagenesis, and our chemistry collaborator with Dr. Junjie Xie and Yu Zhang in Taipei, Taiwan. And the funding is provided by Academia Sinica and Ministry of Science and Technology. And I also thank AMBO for giving me support because I'm part of the EP program. And yeah, and we are recruiting. So if you find our work interesting, feel free to contact me. Thank you.
Thank you very much, Ping, for a very excellent talk. I must say, if carnivorous fungi got the media interested, then using the word fungi that produce nerve gas is going to drive them even more wild. So thank you very much for the talk. So just to remind everyone that the way we work now is we will be going over to Oliver, who will introduce our second speaker, and then we'll have pulled questions at the end. And please remember to use the Q&A function for questions discussed at the end. Over to you, Oliver.
Yeah, thank you very much, Gordon. And it's my pleasure now to introduce Professor Jun Kwon-Chung as our second speaker. I guess Jun can be called a pioneer in medical mycology, and everyone who digs into the field will certainly have come across her work and one or the other cornerstone publication. Jun studied biology in Korea, and then she came to the US and did her PhD at the University of Wisconsin, and then switched to the NIH and NIAID, where she was continuously promoted until she became chief of the molecular microbiology section, and this is a position she still holds today since 1995. Jun has been honored by numerous keynote presentations and awards. I cannot list all of them here without taking away most of her speaking time, but I want to mention that she has been a keynote speaker at one of the first meetings of the European Conference of Medical Mycology in Dresden in former Eastern Germany, and this was one of my first conferences that I ever attended, and I can still remember hearing her talk there. She has received the Lucille George Award, the most prestigious award of the ISHM, our international society, and also in 2017 the ASM Lifetime Achievement Award. And with this, I will stop. I think Cryptococcus has always been at the heart of her work, and she's also going to talk about Cryptococcus today. Jun, the stage is yours.
Sure. Which one? This? Yeah. Is that the first one? First. Okay. Can you hear me?
Yes, perfect.
Now, there are two etiologic agents for cryptococcosis, and I think everyone knows it. And everyone knows how important and serious disease the cryptococcosis is, because it is a fungal disease that kills more people than any other pathogenic fungi. Now, the two species complex of etiologic agents have been considered as one species with two varieties for long time. Now, this one, Cryptococcus neoformans, was the first species identified, clinically isolated from necrotic lesion in German lady who later died by CFS infection. And that was 1894. And Cryptococcus gattii was isolated 1 year after the German case from French man. And looking at the haploid stage, they are indistinguishable, and they cause the same disease, and they're treated the same way. So these two species has been considered as one species for long time, until year 2002, when I separated them as a separate species. Now, these two species are species complex because they have genetically diverse lineages within the species complex. And as you can see, both gattii and neoformans, they all have a different genetic lineage group. And now, why is it's not showing? Now, I think that, how do I erase this part? Now, this disease is caused by inhalation of either spores, sexual spores, or dehydrated yeast cells, which is small enough to get into the alveolar sac. And then when host is immunocompetent, they cannot cause any disease. But if the immunological defect is present in the patient, they will grow in the lung and then cause fungemia, spread by hematogenously, and then enter by crossing the blood-brain barrier to cause CNS infection. Now, cryptococcosis is 100% fatal unless treated, and even if treated, very high percentage will not survive, which is roughly about 30%. Now, since these two species were considered as one, because they are causing the same disease, treated the same way, but as we look into carefully, there are stark differences between these two species. So today, I'm going to focus on how they are different in pathobiology and antiviral immune environment, and the major risk factors for infection, and mechanism of resistance to 5-FC, and then environmental niche. Why are we studying antiviral immune environment? This slide will show you why we were interested. Now, when we look at the environmental isolates, Cryptococcus gattii, blue, and neoformans, red, they are about half and half. So they're as frequent as same. But when we look at the HIV uninfected patients who has cryptococcosis, then they're about 60% due to neoformans and about 40% due to gattii. But when it comes to HIV positive patients, the difference is very stunningly different. gattii is just only a fraction of the whole HIV patients with the cryptococcosis. So our question was, does the antiviral immune environment, which is HIV positive, select against the gattii differently than neoformans? So we used, to study this, we used poly-IC, which was a condensed poly-IC, which we'll call Hiltonol. And that is poly-IC condensed with the lysine and carboxymethylcellulose, which is a double-stranded RNA virus mimic. And this compound, Hiltonol, is in phase 2 clinical trial for cancer treatment. So we use a black 6 mice and then infect the mice 5,000 yeast cells of either neoformans or gattii. And then we dosed poly-IC 5 microgram, 2 doses per mice each week. And then control received the PBS. At the end, we looked at the survival rate, and then between 7 to 21 days, we have done colony-forming unit analysis, facts analysis, and histopathological study. When we use the poly-IC in the first day, we can already see how interferon alpha concentration has increased drastically in the bar that we drew from this mice. And also in the lung tissue, type 1 interferon beta concentration shoot up in both mice which got the neoformans and the gattii treated with the poly-IC. And survival curve definitely shows that poly-IC treated mice live about twice longer, whether it's infected by C. neoformans or C. gattii. And the difference was more clear in the gattii infected mice. And when we look at the histology, and this is mice that was infected by neoformans treated by control PBS, and poly-IC controlled mice in the same day, 21st post-infection. You can see the PBS control has wall-to-wall in the lung with the proliferation of cryptococci. And then poly-IC treated mice, you can see very nice containment. So it's no longer spread all over the place. Now, in mice that infected by gattii, if you look at PBS, now this blue color is Archean blue, which stains acidic capsule. And hugely infected gattii is just packed the whole lung, whereas poly-IC treated, we don't see this containment, but we see the complete reduction in the number of E cells and production of capsule. When we looked at the colony-forming unit, neoformans treated by poly-IC and PBS, there was about half log difference in the lung, but in the brain, there was at least 2 log lower CFU in the brain of mice treated with poly-IC. And in Cryptococcus gattii infected mice, Cryptococcus gattii has a hard time getting out of lung to disseminate to the brain. So you always get something like less than 100 colonies at the day 21. But in the lung, there's at least 1.5 log colony forming units reduction is seen. And we have shown about 3 years ago that poly-IC is sensed by melanoma differentiation associated gene. It's a cytosolic sensor, MDA5. So we were asking whether CD4 T cells are important in protection induced by poly-IC. We used MHC class 2 knockout mice treated with PBS at poly-IC, and this is a wild type mice, PBS and poly-IC. As you can see, the protection once MHC class 2 is lacking, reduced drastically compared to the wild type treated with poly-IC. And whereas Cryptococcus gattii, we didn't see the difference. We still see the protection by poly-IC, but MHC class 2 knockout did not make much difference. And log colony forming unit in the lung, as you can see, that CD4 antibody depleted mice. Now, this is a knockout mice story, but we antibody depleted CD4 and then counted log CFU of Cryptococcus in the lung. And you can see that lung, there is only very small reduction, but in the brain, huge increase in the colony forming unit of neoformans once the CD4 mediated depletion of depletion was done by antibody. However, in Cryptococcus gattii, you don't see any difference whether CD4 induced reduction of CD4 cells by antibodies there or not. You don't see any difference. So question is, type 1 interferon expression due to HIV infection in its very early stage may protect mice from cryptococcosis. At the CD4 depleted later stage, when it's in the patients are in AIDS, they may be greater risk for infection by C. neoformans than C. gattii. If it's not T helper cell mediated immunity, how does poly-IC protect mice from C. gattii infection? So we use beta-2 microglobulin knockout mice to assess the role of CD8 cells. Type 1 interferon expression due to HIV infection in its early stage might protect patients. When depression, when it's AIDS patients, they usually have CD4 cells less than 200, and it's a severe case, it's below 50. Those patients may be at the greater risk for gattii neoformans infection. And so, is CD8, NK cell, and B cell, are they all important for poly-IC mediated protection from gattii? That was our question. As you can see here, we depleted CD8 by 2 different antibody clones, and then NK1 was also depleted by antibody. And then we used the knockout mice, which will knock out T cells and B cells. As you can see here, there is no difference, meaning that cellular immunity does not play any function in the protection of gattii infected mice by poly-IC. So the question is, now this is beta-2 microglobulin treated mice. If you can see, it's completely reduced. Protection, knockout mice was not protected by poly-IC. So then we were asking, what is the function of 2 microglobulin? Microglobulin is a cofactor for MHC class 1, and then antibody recycling, and then transferrin recycling. So knockout mice has a phenotype of CD8 and NK cell function is compromised when we have knockout mice with the beta-2 microglobulin. And also serum antibody deficiency because of the B cell issue. And ion overload, which cause hemochromatosis. These three are very well-known phenomenon with the beta-2 microglobulin. And as we have seen already, the CD8 NK cell really doesn't do anything, and B cell and T cell is not involved in the protection. So what left is ion overload. Now we ask question, is ion overload is causing the protection of mice from gattii infection? Here is, when we supplemented the ion, it abolished the poly-IC protection, and deprivation of chelation mimics poly-IC protection. So obviously the evidence that ion plays a major role in protection was seen here. When we added ferric chloride 3 times a week into a mice which was getting poly-IC, you see there is no difference in terms of colony-forming unit, meaning that no longer it's protected. And this deferoxyrox is a chelating agent of ion. When ion is chelated, the poly-IC effect and chelating agent effect, they all protected, which means ion is the major reason. So when we looked at the poly-IC-induced ion binding protein in BAL, we saw huge increase with the poly-IC treatment. But when MDA5 knockout mice was used, you remember that poly-IC was sensed by MDA5. There's no significant difference. And ferritin also concentration hugely increased in the BAL, but not in the MDA5 knockout mice. So this gave us confidence that ion overload must be the reason why beta-2 microglobulin worked to negate the protection of poly-IC. So if we can summarize, what happens is that in the black 6 mice without poly-IC, the immunity is a Th2 type, and the killing of Cryptococcus is not very efficient. Eventually, CNS dissemination occurs. However, with the poly-IC interferon, type 1 interferon alpha and beta effect will change into Th1 immunity with interferon gamma, TNF alpha, IL-17. So it's a very improved containment, which is seen in this lesion. And unlike in Cryptococcus neoformans, for gattii, when poly-IC is introduced, then ferritin, transferrin, kind of a ion binding proteins leaked out from the blood vessel and then bound to the free ion so that there won't be enough ion for Cryptococcus gattii to grow, whereas without poly-IC, there's enough ion concentration for gattii to cause profound pneumonia. So we were asking, does poly-IC mediate induction of pulmonary vascular permeability? Because there has not been known that poly-IC does that. So we assayed various serum proteins in BAL samples followed by poly-IC dosing, and injected a traceable molecule of phytidextran, which is 70 kilodalton, intravenously assay the amount of phytid that crossed into the lung airspace. And then apply the gene expression analysis of cytokines, chemokines in BAL, as well as use a knockout mice to determine the pathway involved in lung vascular permeability. And the treatment of mice with C4880, which is a non-inducer of vascular leakage, also enhanced the expression of transferrin and other serum proteins and protected mice from gattii as a poly-IC. So this experiment supports our original idea that leakage is the one that caused the ion restriction in the lung that prevented or helped gattii, helped the mice to be protected by poly-IC. So we tested the concentration of blood proteins, serum proteins in the BAL, and transferrin, ferritin, lipocalin, albumin, and this is total protein and complement C3, all serum proteins. When we had one dose on day 0 of poly-IC, you can see huge increase of the concentration of these serum protein in the BAL. It's the same kinetics. We can see all 6, including suggesting that permeability is happening with poly-IC treatment that releases all these proteins into the BAL, and that can restrict the availability of iron. So when we asked whether this is specific for iron binding protein or is this general leakage? So to test that, we use the fluorescein dextran at 70 kilodalton intravenously injected and let these dextran seep into alveoli with the poly-IC treatment. And then we get the bar and isolate and then analyze the concentration of how much of the 70 kilodalton fluorescein dextran is in the bar. So this is our test schedule. The poly-IC aspiration is only day 0. And then this dextran was IV injected every day for many, many days. And then BAL was collected from all these mice. As you can see, in day 1, when poly-IC was dosed, you can see huge increase in the dextran within BAL, meaning that this dextran crossed and came into the alveolar sac. Now, to get there, has to be, permeability has to be increased in the blood vessel. But since there is no poly-IC treatment in the day 2, all this, so you can see that there is no increase in the fluorescein dextran concentration in the blood. Now, we wanted to see how this leakage has happened or permeability increase was happened. So what we did was we used MDA5 knockout mice, which cannot sense the poly-IC, and then PBS with the black 6 mice and MDA5 knockout mice versus poly-IC mice with the MDA knockout mice. And when concentration of cytokines and chemokines that appeared in the BAL was assayed, and then fold increase has been tabulated here. And you can see when MDA5 is knocked out, no longer can sense the poly-IC. We don't see any increase except CCL2 had some expression, but much lower than the poly-IC treated one. So this is the interferon beta, IL-1 beta, CXCL2, and all of these different chemokines and cytokines. Every one of the cases show that MDA5, there's no expression. So we came to the conclusion that leakage happens with the poly-IC, and then this poly-IC will be going to MDA5 dependence pattern to MAVS, and then type 1 interferon will, pro-inflammatory cytokines will be expressed hugely with the type 1 interferon. And when this is expressed, vascular permeability happens. And this is why we concluded that poly-IC is encouraging or enhancing the leakage of vascular wall so that the ion binding proteins are coming out to the BAL. Now, next difference I'd like to talk about is a major risk factor of neoformans and gattii, supposedly in immunocompetent patients. The reason why we studied this is because Cryptococcus neoformans has always been known as an opportunistic pathogen. However, many people still believe that gattii is considered as a primary pathogen because unlike neoformans patients, there are just so many patients who do not show recognizable immunosuppression and yet get infected by gattii. But we believe there must be hidden immunological issue in the patients who are supposed to be healthy otherwise. So we collected 135 global-based cases of gattii infection from 5 continents with a medical history clearly tabulated. And then also we could get the strains to see what kind of molecular type the isolates are. So what we did was the immunosuppressed patients were 49, and then immunocompetent patients were 86, altogether 135 patients samples. But unluckily, plasma samples were only available for 32 because they don't keep the plasma samples. But we were at least fortunate to be able to collect 32 global gattii patients' plasma, and then we could test whether there is a cytokine inhibiting antibodies there. And so when we tested, we previously had known that GM-CSF autoantibody is present in the plasma of the patients who are supposed to be healthy and get the gattii infection. So when the sample was received, they diagnosed as a 29 completely immunocompetent and 3 were immunosuppressed. What that means is 91% is immunocompetent and 9% was immunosuppressed. But when we tested their plasma to see if the plasma can phosphorylate the step 5, which is the mechanism of GM-CSF, and what we saw was that 72% was positive for antibody, and therefore only 28% were negative. But some of this genome sequencing has been shown that there is a mutation, and this mutation may be something to do with hidden immunity, immune suppression in the gattii patients, supposedly healthy or competent gattii patients. So if I summarize the major risk factors, no question, neoformans, HIV is a major risk factor because 75% of patients who get the neoformans are HIV patients. But patients with anti-GM-CSF autoantibody, we only see 10% of them get Cryptococcus neoformans infection, whereas 90% get gattii infection. This tells us that hidden immune defect in supposedly immunocompetent gattii patients has this antibody. Now, how are these two species differ in 5-FC resistant? And we found that there is a nitrogen source is influencing the difference. Now, this is very well studied pathway for 5-FC resistance. Now, 5-FC is a fluorinated pyrimidine, and it comes into the cell by permease FCY2 and then deaminated by FCY1, and eventually it will inhibit the protein synthesis, DNA synthesis, RNA synthesis, and as a result, protein synthesis. So 5-FC is a very useful drug and used in combination with amphotericin B for initial treatment of cryptococcosis patients. But this cannot be used as a monotherapy because there are a huge number of resistant clones appear in a very short time. And because of this phenomenon, they cannot be used as a monotherapy. So we, of course, everybody knew that 5-FC induces this resistant clones, and that's why they cannot be used as monotherapy, but didn't know why, what is the cause of this appearance of resistant clones. And we found out that nitrogen source in growth media affects 5-FC resistance only in gattii, but not in neoformans. So this is a stark difference between the two species. Now, this is like 300 cells were plated on 0.5 microgram of 5-FC, and this is a strain name, neoformans. You can look at the MIC is 0.025. So which means in 20-fold MIC concentration, you still have huge number of clones growing by day 5. And then 40x, and eventually when it comes to 400x, just mutants, which are either FCY1 or FCY2, mutants start growing, but none of these in between resistant clones. And Yun Cheng has analyzed that and published in Nature Communication. And we took out all these clones and then did the analysis for chromosome number variation and sequence, mutated sequence to figure out what these resistant clones are. We saw total chromosome doubling or partial doubling of chromosome 1, or some piece of chromosome was deleted in some resistant clones. So at the chromosomal level, very different effect is given by 5FC to Cryptococcus neoformans. But when we looked at the sequence level, plethora of mutations at the sequence level in both species suggested that 5FC is mutagenic at the moderate concentration, meaning 20 to 100x MIC. And all the wild type were susceptible to 5FC. They had this range of MIC. But every clones that we picked had huge increase. They are resistant to 32 microgram at the 400 to 2000x. Now, up to this point, two species behave the same way because gattii also produce this kind of clones.
June, I'm sorry, just to give you a note that we are running out of time.
Okay, but when it comes to nitrogen source dependent resistance, we saw very different between these two species. You can see on the proline instead of ammonium sulfate, asparagine and glycine, arginine, you can see that FCY2, which is a permease mutant, still is sensitive. So these two species are very different depending on the nitrogen source. So when we found out the primary permease and deaminase are mutated, secondary permease and deaminase, FCY6, function under certain amino acid only in C. gattii. And gattii family transcription factors were found to regulate nitrogen source dependent 5FC resistance in gattii in the absence of ammonium sulfate because it's of nitrogen catabolism repression. Now I will have just one slide for ecological difference between two species. We found in Zambia that Hyrex droppings, we collected Hyrex droppings, actually Dr. Fisher's group collected it and we got the culture. And we found VG5 as a new lineage, only gattii lineage from this ecological niche, but not neoformans. And it is from Zambia, which is Africa. An interesting thing is these strains are very resistant to fluconazole. And this is a concerning phenomenon because, you know, the AIDS patients are treated by fluconazole in Africa. So I'm going to finish by showing my outside of NIH, these people who collaborated on these different projects, and then our section, and then other part of the NIH section. Thank you. I'm sorry that I'm going.
No, perfectly all right. Thank you very much, June. Can you perhaps stop sharing your screen so that we can all return back for the discussion?
Yes.
And I'll hand over to Gordon for the first questions that go to Ping.
Thank you very much. Thank you very much, June, for your presentation. Ping, do you want to turn on your camera so you can join us? There you are. So Peng, so there's been quite a few questions that have come in, which
Sure.
I've now lost. There they are again. But I want to take the chair's prerogative and just ask a question because I didn't quite understand your model. So you've got a pore, right, or a ciliated pore that allows your nerve gas to get into the nematodes. Is that right?
Mhm.
And then
Yeah.
is that toxin then itself distributing through the worm, or is it the toxin is triggering a inflammatory the inflammatory cascade or the calcium cascade through the worm that's causing it? I didn't quite understand your model there.
Yeah, okay. So we do not have the direct evidence to show that the toxin itself can diffuse throughout the entire nematode, okay? But then we do have the evidence that, for example, like the wave of the propagation of the calcium is related to the dosage. So it's dosage dependent. So if the nematode touch just one single toxocyst, okay, then the wave doesn't travel. But if it contact more toxin, then we observe that there's a propagation of the calcium wave.
The concentration dependent.
So it's concentration dependent. Yeah.
Okay.
So it does disrupt, like once it contact the membrane, we think that it just disrupt the membrane integrity. And then at least like in the head region, that's direct contributing to the damage of the cells. Yeah. Mm-hmm.
Okay, back to you, Oliver. Thank you.
Right, so we have some questions for you, June, and also can I ask you to turn on the camera again? And the first question is here about the effect you described on T-cell immunity and the two Cryptococcus species. Do you think it's possible that Cryptococcus gattii suppresses the CD4 response kind of actively?
I didn't get it.
The question is, do you think that Cryptococcus gattii actively suppresses the CD4 response?
No, I don't think so. I don't think we have that kind of evidence.
Okay.
So whether CD4 is there or not, gattii doesn't care because iron is the issue, but gattii does not really act on the cell immunology.
Okay, so the additional suppression of the antibody response is independent of the CD4.
Yes, I think so.
Okay. And then I have another very interesting question, and this is about the effect of the poly-IC that you described on vascular permeability.
Right.
Do you think that this is an effect that could also be important for other fungal infections that occur in the presence of viral infections like aspergillosis, influenza, or COVID?
I guess so, because it's a acute respiratory disease of lung disease. This may be a nutritional immunity that will work for other pathogens in addition to C. gattii. So maybe other virus, bacteria, or fungal, but those areas we completely don't know.
Okay, and then I have a question here about the autoantibodies against GM-CSF. Has this been studied in other patient samples with a broader range of fungal infections or other types of Cryptococcus infection?
Well, GM-CSF autoantibody study was done because when we had all this plasma collected by Dr. Bennett for years and years, and we were assaying, and those people because Dr. Bennett collected the Cryptococcus from patients, and we noticed that somehow that GM-CSF positive plasma the patients are the one who are infected by gattii, not neoformans. So that gave us an idea that maybe gattii is not a primary pathogen, but has a hidden immune defect because the laboratory doesn't test anti-cytokine antibody for paneling or profiling immunity of patients. So it remains as a hidden.
Okay.
But we find that nocardia, cryptococcus, this kind of a pathogen is associated with the GM-CSF autoantibody.
Okay, so there is a broader effect of these autoantibodies.
Right.
Okay, thank you very much, Gordon. Back to you.
Thanks very much, Oliver. So Ping, some more questions. I'm going to try and group them just very slightly. So there's a question about the actual 3-octanone. The first one is dosage effect. I think you've answered that question. Have you tested this compound on any other worm species?
Yes, it basically paralyzed like all the worm we tested.
Okay, so it targets membranes, and is this only specific to membranes of worms, or does it affect other eukaryotic cells? We eat this a lot. Why are we not dying for it?
It affects like basically all the eukaryotic cells we tested. So so it's really concentration dependent. So if the concentration is high enough, it paralyzes like lots of things.
So if we eat enough mushrooms, is that what you're saying? We eat enough of these oyster mushrooms. It's not good for us.
Well, but this structure is only on the fungal hyphae, but not on the mushroom itself. So mushrooms, the fruiting body.
Right. Okay.
So it doesn't have list toxosis. So we only observe list toxosis develop on the mushroom hyphae, and it's also only when the hyphae was grown under nutrient starvation condition. So if there's like a nutrient-rich environment, they don't develop this structure.
Okay, so presumably when they're culturing it for human use, it's on enriched media anyway, so that wouldn't develop. But one of the questions that came in was about that structure, the toxicity structure, was to ask, in your mutant selection, right? Did you find one that produced the cyst but didn't have the toxin?
Yeah, we do have like a couple mutants. Okay, they develop like a very few number of the toxocyst. So so the mutant that completely lost the ability to paralyze worm, they basically do not develop toxocyst at all. But we do have some mutant, okay, when we test the, like how good they are in terms of their ability to paralyze C. elegans, they still maintain certain level of paralysis activity. And when we look at those mutant, okay, it's that they can still develop coccosis, but the number is very little.
This number is low. So is this a secondary metabolite, this 3-octo, whatever it is, octanone?
Octanone, yes.
So presumably I guess what the question really is getting at, is this toxicity structure intimately linked with the biochemical pathway, or is it a completely separate?
Right. Yeah, we do not know yet now, okay, because we do not know like exactly what's the pathway that regulate the 3-octanone biosynthesis, but presumably like maybe some of our mutant could have a defect in making this compound.
Yeah, so
yeah, right. And we don't know the answer yet, but I think if the cells cannot make this compound, the structure wouldn't probably won't develop to full extent.
Okay.
Mm-hmm.
And then the last question that I have for you that was posed by the audience is, what receptor on mitochondria leads to the influx of calcium?
So we have tested like a couple calcium channel on the mitochondria in C. elegans. Okay, so basically it doesn't completely block the calcium influx, but it does reduce the calcium level.
Okay, so there's a specific channel, or is it a group of channels?
It's a calcium channel on the mitochondria membrane.
Okay. So it's the one specific molecule that you're targeting there.
Right, like so we tested like the mutant
Okay.
the calcium mutant and measure the calcium influx in that particular mutant. Yes.
Okay, thanks very much, Ping. I'll hand you back to Oliver.
Okay.
Yeah, thank you, Jun. I have one question here. So I was very interested in the central role of MDA5, but what about other receptors that may recognize double-stranded RNA, like toll-like receptor 3 or something? Are they also involved in this, or is this completely independent?
I don't know, because we have not tested about that.
But from the data you showed, you would basically conclude that you can link back most of the effects to MDA5.
Oh, yes, MDA5 senses this poly-IC. That's why it's almost like you're knocking out the receptor for the poly-IC.
Okay, so the sensing of poly-IC is specific for MDA5 and not for the other double-stranded RNA receptors, is it?
You know, just the plain poly-IC is sensed by TLR3, but this one is condensed one to make it more stable, and then the receptor sensor was different, MDA5. So I'm sure MDA5 senses other things of DNA and nucleic acid.
Okay, right. So thank you very much. I think we've made it to answer all questions in the Q&A, and knowing how late it is in Taiwan, I think we need to finish up now. Thank you very much to both speakers for excellent talks and excellent discussion. And to the audience, please remember next time for Mycotalks is the 25th of May. Thank you very much and enjoy the rest of your day.
Thank you, everyone. Thank you, June. Thank you, Ping.
Okay, thank you.
Bye-bye.
Bye.